Hello my schoolers, you're welcome to my school YouTube channel. My name is Emmanuel and in today's video we'll be tackling JAMB past questions in biology for the year 2001. Please don't go anywhere, stay with us and we'll be right back. You're welcome back to my school YouTube channel. Today we'll be tackling questions from number 1 to number 25. Now let's get down to question number 1. An association between the root nodule of a leguminous plant and rhizobium species is known as what? First, we must understand the relationship that exists between leguminous plants as well as the rhizobium species. The rhizobium species actually is a bacteria, okay? That is why if you look at the options here, this actually could have been the answer because we have mycorrhiza. Mycorrhiza is a relationship that exists between the root nodule of a plant and fungi, okay? Of course, both of them will benefit from each other, from that relationship. But because this is a relationship that exists between the root nodule of the plant as well as the fungi, okay? This will not be the answer because it's leguminous plant and rhizobium species. This is a bacteria, not a fungi. So option B will not be the answer. So what happens is that the leguminous plant actually, needs atmospheric nitrogen to ensure the growth and the development of the plant. But the atmospheric nitrogen that is provided is not useful directly to the plant. So there is need for that nitrogen to be converted to useful form for the plant. So that is why the rhizobium species will attack the leguminous plant. Once that attack is made, as it attacks it, it will now cause a nitrogen fixing nodule. It will produce that. So when it does that, this will now be able to convert the atmospheric nitrogen, okay, that the plant needs actually into ammonia, okay. In such manner, the nitrogen is now being produced for this to get. So in that way, this has benefited. Then on the other hand, this rhizobium species is also getting nutrients from this leguminous plant, okay, because it needs to make its food. It makes it from the succinate, okay, the malate. Those are the food form in which the rhizobium species, the bacteria, actually needs this. So in that manner, both of them benefit from each other. So it's a symbiotic kind of thing, okay. Both of them, there is no harm. If you look at commensalism, the commensalism relationship is a relationship where one of them is benefiting while the other one is not benefiting at all. No profit, no benefit, no harm, okay. So it is not this. I have explained this before. It is not this because the relationship exists. This one is an association that exists between the root of the plant, okay, and then a fungi, okay. So this is not, because this is a bacteria. Option C, parasitism, has to do with the kind of relationship where there is, of course, there is one that is causing harm. One is benefiting. As it's benefiting, the other organism is causing harm on the other one, okay, on the host. So it is not this. So symbiosis has to do with a relationship where two species or organisms actually benefit from each other. They both have benefits from each other. So that is why the correct answer or the best option here is option D, symbiosis. Question number 2. Amphibians are normally found where? Of course, amphibians are organisms that live both on land, okay, and water. Not just on land, on moist land, okay. The land, if it is a dry land or arid land, you won't see them, you won't find them there. They live more on moist land. So because of the structure, the physiological and the morphological nature of their body system, okay, they need a moist environment or habitat. So that's why you find them in the water, live in water, and they also live on land, like your frog, your salamander, your toad. Okay, we have those organisms that live at that. So amphibians are normally found on dry land and water. In water, yes, but not on dry land, okay. Option B, in water and on moist land. Yes, the environment has to be moistened, okay. Option C, on moist land. Not just on moist land, they also live in water, okay, in water. So not only the organisms. So the correct answer here is option B. Amphibians are normally found in water and on moist land. Option B. Now let's move on to question number 3. Viviparity occurs mainly in the where? Is it in mammals? Is it in reptiles? Is it in apes? Is it in amphibians? Okay. Viviparity actually is a type of reproduction where an organism, there's fertilization that takes place inside of the organism, and then the baby is actually born in the womb, and then it's giving birth to a life. Okay, that is where we have viviparity. Another one that is closer to this, that is common, is oviparity. Oviparity has to do with organisms that have their offspring born in eggs. Okay, so they lay those eggs, and then after some time they lay that. So viviparity, the organism bear the offspring, the fetus, it develops in the womb, and then it grows up into an embryo. Okay, inside, then it's giving birth to a life. So among these options now, amphibians do not. Of course, we have some of them that actually give birth to their young ones, okay, alive, but most of them actually lay eggs. Okay, then we have apes. Okay, we have apes, actually like the birds. Okay, they lay eggs. Okay, in having that. So it is not that. Options B is reptiles. We have some reptiles that lay eggs, like the lizards. Okay, yes, we have some of them that give birth to the young ones alive, but because we have some that actually lay eggs, so this will not be. But mammals, I don't know if you have heard of any mammal, any human being, okay, laying egg before. Of course, it's going to be bizarre. It's going to be so strange. Okay, so it is option A. Viviparity occurs mainly in the mammals. All mammals give birth to their young ones alive, and they breastfeed them. Okay, because they have mammary glands. Okay, like we have that. So option A is the correct answer. Question number 4. The jointed structure in insects that bears organs which are sensitive to touch, smell, and vibration is the what? We have the maxilla, we have the labium, we have the antenna, we have the abdomen. Okay, this one is in the outermost part. Okay, yes, we have it in the thorax. From the thorax region of the insect, you have it down. Okay, this would have been the answer if it was just sensitive to touch. Okay, but this, for the smell, this doesn't work. So vibrations, yes, but for smell, the abdomen cannot smell. Okay, for the insect. So option D is not the answer. We have antennae. Okay, antenna has jointed segments. Okay, jointed segments that are jointed together. Okay, before we have it, of course, we have them on insects. Insects have antenna. The antenna, usually they are in pairs. We have 2 of them. Maybe in mosquitoes. Maybe you have it in housefly. Maybe you have it in butterflies. Okay, maybe you have them. Most insects, okay, have that antenna, a pair of antenna. So what they do is that once that antenna touches any point, they know that they are so sensitive to it that once there's any touch, they feel it. Okay, it also smells. Okay, then when there's vibration, the antenna makes some senses that the antenna will definitely make on the insect. So that's part of the insect that is responsible for sensing. Okay, the sense, for vibration, for smelling, okay, is the antenna. Okay, we have the labium here, we have the maxilla part of their mouth part. Okay, that can be used maybe for vibrations, okay, but not definitely for touch and then for smell. So the correct answer here is option C, the antenna. The antenna is the jointed structure in insects that bears organs that are sensitive to touch, sensitive to smell, and sensitive to vibration. So the correct answer is option C, antenna. Question number 5. Which of the following groups is the most advanced? We have the pteridophytes, we have the bryophytes, we have the talophytes, we have the gymnosperm. Okay, we have them. Of course, there's the last one that is the angiosperm. All of these ones are the groups that we have underneath the plant kingdom. Okay, so but there's a level of stages of advancement. Some are more advanced than others. What are the criteria we use in checking for the advancement? We look at their vascular bundles. Okay, do they have vascular tissues present in them? They have seed formation. Okay, we look at them. Are they born inside? Do they bear seeds? Do they have those ones? Do they have it? So all of those things we look at. Their body differentiation. Do they have the roots differentiated from the stem, or they are all together? So all of those things are criteria we use in checking for the advancement of the plant groups. Okay, so we have them usually here. There is a mnemonic usually that I use, okay, that help me, that guide me in order of the advancement of this plant kingdom. Let's look at the board and see. Okay, in order of advancements, okay, we have T, we have B, we have P, we have G, we have A. Usually what I do is I call this, I will say Tabitha, you can put any name there. Tabitha buys potatoes and granules always. Tabitha buys potato and granules always. So that is for me. You can form yours, okay? So this is the level of what? Advancement. So the Tabitha, the T is actually the thallophytes. Okay, we have the thallophytes, we have the bryophytes. For B, we have the pteridophytes. Here we have the, for the granite, yes, that's the gymnosperm. Okay, and then we have the angiosperm. Angiosperm is the most advanced among them, okay, among these 5. So this is the list, followed by this, followed by this, followed by this, and then followed by this. But if you look at the options, we don't have angiosperm there. So we have thallophyte, we have bryophyte, we have pteridophyte, we have gymnosperm. Actually, the thallophytes do not have differentiated body structure. Their body structure is not differentiated, just like the bryophyte. Okay, the thallophyte, no body differentiation. Okay, that's the root and the stem. It's not differentiated, separated from themselves. Okay, then they don't have vascular bundles, so no vascular, so they are non-vascular bundles or tissues. Okay, the same thing with bryophyte. Bryophyte also, they don't have vascular, so they are also non-vascular bundles. Okay, but the bryophyte, the gymnosperm and the angiosperm, these ones are the what are vascular. They have vascular bundles in them. So a question came a particular year, still in JAMB, where they asked that which one, they listed some specimen and they asked which ones are the non-vascular bundles. So if you know that thallophytes and the bryophyte, for instance, when we talk about the bryophytes now, usually we talk about the liverworts, we talk about the mosses, okay? Those are examples of the bryophytes. So they are non-vascular, but this one do not have differentiated body, okay, system, but this one have their body system differentiated, okay? But this one, both of them actually, they are non-vascular bundles, like I said earlier, and then they don't also form seeds, okay, in their fruit, both of them, okay? But for these 3, this one have differentiated body system, this one has their vascular bundles, okay, but this one do not have seed formation in them. That's the pteridophytes, okay? The gymnosperm actually have, they are also vascular bundles, they have differentiated bodies, okay, body structure, and then they also bear fruits, okay? But their fruits, the seeds they bear in their fruits are not within the fruit, okay? They are in the outermost. They are not enclosed in the fruit. That's the difference between the gymnosperm and the angiosperm. The angiosperm bears seeds also, okay, but their seeds are enclosed inside of the fruit. So that's the difference between them. In other words, now, for the sake of our question, in order of advancement, this is the list, followed by this, followed by this, followed by this, followed by this. Well, from the options, we have these 4 of them. So gymnosperm actually is the highest. When we talk about in terms of advancement, is the most advanced among them. Don't forget, Tabitha buys potato and groundnut always. Let's move to the screen now and see what we have here. Okay, so which of them? We have the pteridophyte, no, we have the bryophyte, we have the thallophyte, and we have the gymnosperm. So the correct answer here, the most advanced for what we just did now, is gymnosperm, option D. Now question number 6. Most monocots are easily recognized by their what? Don't forget that monocots are actually plants, okay, that have a single monocot. Mono is one, okay, a single kind of leaf, and the seed leaf, okay, that they have. So how do we recognize them easily? Option A says short leaves with petioles, okay. Petioles, yes, they have, but they don't have the short leaves are not there. You see, they are not. For instance, if you look at the maize, okay, maize, when you see a maize plant, you see how long the leaf is. So the leaf of monocots are actually long, not short, okay. And then you see that on both sides of the leaf, they are sharp, okay, like a sword. You touch it, it's as if it wants to cut you. On this other side too, it's not like, you know how cutlass look like, that only one side is sharp. But for monocots, they have long leaves, and then you have, you know, those two are sharp like sword, okay, the 2 sides of the leaf. So we say short leaves with petioles. No, this is not correct. Option B says long and sword-like leaves. Yes. Option C, long and palm-like leaves. No. Okay, they are sword-like, not palm-like. Like we have those ones that have a seed like the palm tree and all of those. No, but like I gave you an example of monocots, you have those ones, they have their veins in the leaf, okay, like venation, like not like net. You see those ones, the net like veins are like those ones you can see in dicot. So option C is not the correct answer. Option D is not. We are short with veinlets. They are not short, they are actually long. So the correct answer here is option B, long and sword-like leaves. Question number 7. Water fleas, wood lice, and barnacles belong to the group. What group do they belong to? Don't forget that these organisms, actually you can find them in water, okay, in moist environment, okay, like the other examples. We have the lobsters, we have the crabs, okay, we have the prawns, okay. They are all example of a group of organisms that belong to a group of Animalia, and that is called the crustaceans. Okay, they are all called crustaceans, not the arachnida. The arachnida are the spider, the scorpions, okay. Yes, those ones fall and underneath that, so they are not. Water fleas, you can find them water, the wood lice, and then the barnacles. Like I gave you other example, the lobsters, the prawns, the crabs, they are all crustaceans, okay. These are crustacea, okay. Insecta, no, they are not insects, okay. Maybe if you see flea, you might be feeling maybe it's an insect. No, the wood lice and then the barnacle. These ones are just to confuse you. When you see lice, you say, oh lice, maybe that's, other lice that flies and this. But they have specific characteristics that categorize them to be under the group of crustacea. Then we have the chilopoda. So the correct answer here is option B, crustacea. Now let's move on to question number 8. The mode of feeding in amoeba, some people call it amoeba, okay, but that's the right pronunciation is amoeba, okay. The mode of feeding in amoeba and hydra is what? Is it heterotrophic? Is it holophytic? Is it autotrophic? Is it symbiotic? So the question you need to ask first is, are they photosynthetic plants? Okay, the amoeba is not a plant because it can move from one place to the other. You see it on this, and then they feed also other organisms. So they are animal-like in feature, okay, because we have the kingdom Protista, okay, so protist. So we have them, and then the hydra. Hydra also have a basal foot, okay, which move. It has gullet, it has mouth, so like animal, okay. So they are not plants. So you cannot say that they are autotrophic, because autotrophic, they will be autotrophic if they are plants and they have chloroplasts, and then they can manufacture their own food by themselves using the CO2 and then water to manufacture their food. So option C is wrong. You can eliminate that with the elimination method. Option C is not, okay. Option D, symbiotic. If it is symbiotic, that means both of them are cohabiting together and then they are deriving benefit from each other, okay. But that is not the case here. So option D is also eliminated out. So we have between option A and option B now the elimination method. Now holophytic and autotrophic are the same. They are saying the same thing because both of them are to do what? They are to manufacture. It's a mode of feeding where organisms produce their food, manufacture their own food by themselves, not depending on other organisms for their food, but they manufacture their food by themselves. So option B, option C, you are saying the same thing, okay. But option A, heterotrophic, okay. Heterotrophic organisms are organisms that actually depend on other organisms, okay, like small organisms to feed, okay, to produce their food, or on other plants. So both of them, they have some micro organisms, some small microatoms, okay, like let's say small organisms that they feed on, both amoeba and hydra. So they are not manufacturing their food themselves. They have to depend on other smaller organisms to make their own food. So based on that, option A, heterotrophic mode of feeding, is the correct answer. Question number 9. Which of the following organisms does not exist as a single free living cell? As a single free living cell. Majorly, we have 3 forms, different forms in which living organisms can exist. You can have them existing as single form, or what we call free living okay, free living form. Okay, we have those organisms that exist in filaments, okay, like the Spirogyra, okay, they exist as filaments, and then those ones that exist as colony, they exist in colony. We have them in much like that, like the Volvox is an example of organism that exists in. Now the question is, which of the following organism does not exist as a single free living cell? So which means that among these four options now, one of them does not exist as single, so why the other three exist as a single free living cell. So if you are not sure of, let's do elimination method again. We have paramecium, we have mold, we have amoeba, we have chlamydomonas. Okay, of course we are familiar with amoeba. Amoeba exists as a single unit because it's a unicellular organism. Okay, it can exist, all the functions of the cell are conducted by one. They don't have specialized body, okay, that carry out different so it exists as a single free living organism. Paramecium is another one, okay, that exists like that. If you see the diagram, like a foot, okay, human foot, the foot of human, when you input it on this, and that's how the paramecium goes. Like we have the cilia all around it like that. So paramecium is also a unicellular organism which is existing as a single free living cell. Then we have the chlamydomonas also existing as a unicellular organism because it exists as a free living. If we have Spirogyra exists in filaments, okay. Volvox exists in colony, okay. But from the options we have here, the mold actually is a multicellular organism, okay. This mold is different from the slime mold. The slime mold and this mold, they are different, okay. The slime mold actually is a unicellular organism, you have it there, but for this mold is multicellular organism. So because it's a multicellular organism, it does not exist as a single free living cell, rather it exists as a multicellular organism. So option A, paramecium, is a unicellular organism, it exists as a single free living cell. Amoeba exists as a free living cell, okay, and then chlamydomonas also exists as a single free living cell. But mold, option B, does not. So the correct answer is option B, mold. Do you know you can take practice questions in our simulated JAMB CBT past questions? All you need to do is to click on the link in the description below that takes you to the Myschool website. There you'll be able to download the Myschool mobile app on your Android and then the Myschool software on your laptops and computers. Please go ahead and start practicing. Now let's move on to question number 10. The center for learning and memory in the human brain is the what? We have the medulla oblongata, we have cerebellum, we have cerebrum, we have olfactory lobe. Okay, now medulla oblongata, usually where is it found? It is found in the brain, okay, but at the base, the bottom part of the brain, okay, where it connects. In fact, it connects both the spinal cord. It is the medulla oblongata that connects both the spinal cord, okay, to the other part of the brain. Connect both the brain together and then with the spinal cord. So because of that, it is responsible for heartbeat, okay, for heartbeat, for blood pressure. That is what the medulla oblongata does. It's responsible for the heartbeat and then for posture. But then for the cerebellum of course is also found in the brain, the head. Okay, what this does is that it's responsible for the posture, okay, the positioning, the posture cerebellum does. The cerebrum is the largest part of the brain. The largest, so big, largest part of the brain. And what does it do? It helps for keeping memory. You've read, you are preparing for an exam, for you to be able to what? To recollect what you have read, okay, in the exam. An event has happened, the ability to recollect, the memory, to have the memory, it is actually cerebrum, okay, that is responsible for that. Of course, you see, they look almost alike, cerebellum and cerebrum, okay. Cerebrum is responsible for memory, keeping of memory and then remembering. Then the olfactory lobe is actually responsible for sensation of the smelling because it's connected to the what? To the nose, to the nostrils, okay. So for smell, ability to smell, it is the part of the brain that is actually responsible for that. So the question says the center for learning and memory in the human brain is the cerebrum, okay. Cerebrum is the correct answer. So the correct answer is option C. I hope you are enjoying this content. If yes, please don't forget to click on the like button, hit the subscribe button, and finally tap the notification bell to keep you informed once we upload our next video. Question number 11. Urea formation occurs in the where? This question you have to be careful. Urea formation, okay, not the excretion, okay, not the production. Not the production. We have urea formation. Where is urea formed? Okay, is it in the heart? Is it in the liver? Is it in the lung? Is it in the kidney? Okay, of course. Elimination method, if you know what the heart does, the heart just pumps in oxygen, okay, and then the oxygenated blood in and out. That's what it does. And then the food that are done, that's what the heart actually does. The liver, what the liver does is to form that urea. Okay, there's a conversion actually that takes place. Okay, urea that is formed, it is actually done in the liver. The lung is responsible for excretion. Okay, for respiration, the lungs is what we use for that, but certainly not for the urea formation. Kidney, yes, kidney is responsible for detoxification, okay, for excreting out or passing out the urea, okay, for, of course, it's converted into urine, okay, that is passed out of humans. So the kidney is actually responsible for that, for osmoregulation, for detoxification, okay, for excreting out nitrogenous waste that are in the body, okay, and excess water. It does the balancing, that homeostasis, that's what we call that, regulation of internal body environment. So the kidney does that. So the kidney and the liver happens to be the closest answer because lungs and heart are eliminated, option B and option D. But we have said that when it comes to excreting, okay, movement, okay, is actually done in the kidney, of the urea is done in the kidney, but the formation of urea is actually done in the B. So don't get it twisted going for option D because it looks like that. This one we excrete out the urea as urine, okay, but the liver actually does the formation. The formation is done of urea is done in the liver. So the correct answer is option B. Now let's move on to question number 12. The structure that holds the yolk in position is labeled what? Of course, if you look at this diagram, you can tell. You should identify the diagram first. And that is what? This is the egg, okay? Constitutional area of the egg. So you should be able to identify what each of the parts is. The part labeled I, that's the albumin. Actually, that's albumin. Then the outermost part here, of course, that's the shell. That's the first part you get to in the egg. That's II. Then I. These are like strings, okay? That is holding this. This is the yolk that you have here at the center. When you boil it, you have it as color yellow or light, yellow of that. Okay, that's your yolk there. Okay, and then this is the chalaza. We call it chalaza. Chalaza holds on two sides. You can see it on this side. This is the air space. Okay, this is the chalaza that holds the yolk firmly at the center of the egg yolk. Okay, so this is the yolk, this is the chalaza, I Roman figure 3, and then we have a round stuff at the middle of the yolk. That is the germinal disc. Okay, we call it germinal disc. So that is what this option IV is. So the question says, of course, it's glaring. So now we can be able to identify each of the parts. It says the structure that holds the yolk, which is this, in position, this part now at the yolk. So what is that? That is the chalaza. But they are not asking us of the name, they just ask us for the Roman figure. So you can see it, that this one is the Roman figure. II is the correct answer. So you look at this, of course, it is not the shell that is holding the yolk. It is not the albumin that is holding the yolk. It is not the disc, germinal disc, that is holding the yolk, but it is this one, these strings that we have, a string-like thing, which is III. So which is option D. So the correct answer is option D. Now, question number 13 is still the same diagram from question 12 that was brought forward. Okay, there's a question that we have identified each of the parts already. It says the part labeled IV is the what? IV. I already answered that while I was explaining before. Okay, they say albumin is Roman figure 1. Okay, then germinal disc. Yes, germinal disc is that you can see it like a disc, round like that, inside of the yolk. That's why we have the germinal disc. So that is IV, Roman figure IV. That is B. Option C is the yolk. That's the round one itself. Okay. And option D is the air space. Air space is this part here. You can see the space here that allows it to be here. So, of course, the correct answer that the part labeled IV is the germinal disc. So, option B is the correct answer. Question 14. The gas produced during tissue respiration can be identified by using what? The first question you need to ask is that what gas is produced during tissue respiration? During tissue respiration, usually we have the food that is consumed, okay, You can have it as your carbohydrate, okay, that is undergoing aerobic respiration. That means react with oxygen, okay, to form CO2, that is carbon dioxide or carbon dioxide, and then we have water and then energy that is being formed. That is the reaction, okay. I might not be able to write it now, but food, this is your C6. We can have C6H12O6, okay, plus 6 moles of oxygen, O2, will give, you have an arrow, 6 moles of CO2, carbon dioxide, plus 6 moles of H2O, and then plus energy. Of course, energy is always given off in respiration in form of and that is what? ATP. Okay, that's the form in which energy is produced in humans. So the gas produced from that reaction now is that what? CO2 is actually formed in that. Let's look at the board so that I can see. C6H12O6 react with 6 moles of oxygen, okay, to form 6 moles of CO2 plus 6 moles of H2O plus energy. Okay, energy is given of course in form of what? ATP. Okay, so this is the reaction that takes place in the tissue respiration. Of course, this is aerobic respiration. So if you look at this now, this is the product we have. These are the products that we have. This is gaseous. So this is the gas that is produced, which is CO2. Okay, now let's go back to the screen now. You say the gas produced. Now we know the gas to be CO2 can be identified. How do you identify CO2? Carbon dioxide can be identified by bringing that gas closer to what we call lime water. Okay, when you bring it closer to lime water, the lime water will turn to a milky color. There will be a color change, milky color. That's a confirmatory test for carbon dioxide, CO2. Bring the gas that is evolved closer to lime water, and then you have the color of the lime water will change to a milky color, okay, from what it was. But that lime water actually is one of these. So if you know it, it is a salt of calcium, okay. So salt of calcium, not copper sulfate, not sodium hydroxide, not calcium carbonate. Calcium carbonate is your limestone, okay, which is not. But calcium hydroxide, Ca in bracket OH2, okay, that is your lime water. So in other words, they want to know, if the examiner wants to know if you know the other name that lime water is being called. So react CO2 with lime water, it will change the color to milky color. So your lime water is calcium hydroxide. When you bring it, the color will change. You can be able to identify that, oh, CO2 was actually what evolved or given up in the process of the tissue respiration. So the correct answer is option A, calcium hydroxide. Question number 15. A seedling grown in the dark is likely to be what? A seedling grown in the dark. Okay, that means it does not have access to light. Indigo, what is likely to happen to that? Now, what the light does, don't forget that light is needed for photosynthesis of the plant. Okay, light is also needed for nitrogen. Okay, because we have it, because it's in the dark. Okay, the darkness will not allow in for the vitamins that are needed for the growth of the plant, of the seedling as it is growing. Because it needs nitrogen, it needs some other nutrients that will be responsible for the growth of that seedling. Once the light is not available, it is in the dark, what happens? You discover that the seedling as it's going begins to be pale in color and then becoming, pale, and then losing its because there's no dark, there's no light in that part, so it is darkness. So we have option A says etiolated, okay, which is the right answer for that. So if you know what etiolation is or etiolated, you know that that is the correct answer. So it's a tendency of the plant going pale, okay, and then going like when something is looking malnourished, okay, it is what? Etiolated. And because the reason is because it is growing in the dark, okay. It is going not because it lacks other nutrients or not because there's no, but because it is going in the dark. So the plant becomes pale as a result of that. So that is etiolated. Dormant, no. When you say dormant is meaning that it is going, it just remains static. There's no significant improvement, development of that growth. So it is not sturdy, no, as if it is just strong and then waxing. So that is not what it is. And then we have stunted. When it is stunted, you see it coming up. Yes, stunted. I'll be looking like this, but it's not going to look pale. Okay, so that is why option A is the correct answer. So a seedling grown in the dark is likely to be ethylated. Option A is the correct answer. Question 16. The enzyme invertase will hydrolyze sucrose to give what? So you need to ask yourself, what kind of sugar is sucrose? Sucrose is a disaccharide that is formed from one molecule of glucose reacting with one molecule of fructose. Reacting together will give us sucrose. Of course, that is the sugar we consume. So the enzyme invertase, there's an enzyme actually that makes that reaction to occur. Invertase will hydrolyze. When it's hydrolyzing sucrose, okay, it's going to break down sucrose into the two monosaccharide, the two units that actually combine together to form that sucrose. So let's look at the board and see what happens here. Usually, glucose is a monosaccharide, reacts with another glucose to form what we call maltose. Maltose is a disaccharide, okay? Then we have glucose also, one molecule of glucose reacting with fructose, okay, to form what we call sucrose. And then we have glucose again reacting with galactose. Galactose is also another example of a monosaccharide to form lactose, okay? Now, these three actually are disaccharides. Okay? Disaccharides, while every of these ones, they are monosaccharides. One molecule of this reacts with this. So now, we are talking about what? Hydrolysis of what? To hydrolyze sucrose. When you hydrolyze sucrose, you will get these two back, okay? If you hydrolyze maltose, you will get two molecules of glucose back. If you hydrolyze lactose, which is the milk sugar, you get galactose and then glucose back. But for the sake of our question, we ask that when the enzyme invertase hydrolyzes the sucrose, you will get these two back. So which is glucose and fructose. Glucose and fructose. So let's go back to the board, to the screen, I meant to say. The enzyme invertase will hydrolyze sucrose to give what? Maltose and glucose. No, maltose is a disaccharide, just like sucrose. Glucose is correct, but this is not correct. Glycerol and fatty acid, no. Okay? Glycerol is, that's a propan 1,2,3-triol. It's one of the products of, when you hydrolyze fatty acids, fats and oil. Okay, so we have it with fatty acids. It's not, we have glucose and fructose. Yes, this is for what we just did on the board. And then we have mannose and galactose. These ones are also monosaccharide like this galactose, but it is not. When you hydrolyze sucrose, we will get back what actually formed the sucrose, which is glucose and fructose. So the correct answer is option C. Now let's move on to question number 17. When yeast respire anaerobically, okay, anaerobically means it respire in the absence of oxygen, in the absence of air, okay? So that's anaerobic. So when yeast respire anaerobically, it converts simple sugar to carbon 4 oxide and what? Carbon 4 oxide and what? Don't forget that yeast is a microorganism, okay, that undergoes fermentation. So when you get your sugar or your sugar, okay, reacted with yeast, okay, and then you had water, eventually it ferments, okay. When it ferments, what happens? We have that sugar being converted, okay, because it is fermented. No, once it ferments and then you taste it, after some days you begin to see that it becomes intoxicated because we have alcohol present in it. So once it has fermented, alcohol is there. So with that, we can answer the question that when yeast respire anaerobically, it converts simple sugar to carbon 4 oxide and what? Not oxygen. No. Acid. No. Alcohol. Yes. Water. No. So the two products that we have when yeast, we call it fermentation. This reaction is a fermentation process. Okay. It is used in the brewery industries. Okay. Now, in production of those, the brewery drink, the alcohol, the spirited drink. So, there is fermentation by yeast, okay, on the sugar. The sugar is being fermented to produce carbon 4 oxide and ethanol, or what we call alcohol. So, the correct answer is option C. Now, let's move on to question number 18. The transportation of oxygen and carbon 4 oxide in mammals is carried out by what? We have leukocytes, we have thrombocytes, we have phagocytes, and then we have the erythrocytes. Of course, these are the other names that the blood cells in humans, in animals are called. Yes, the leukocytes are also called white blood cell, WBC. Yes, okay. White blood cells, what they do is to act as the soldiers of the body. Okay, they are the ones that fight diseases. They fight sicknesses that wants to come. Organisms want to invade the system. So the white blood cells are actually there as soldiers to withstand them and fight them. In other words, they are the ones that equip and enhance the immunity of the system of organisms. So this is not the answer. Thrombocytes are also called platelets. Okay, that's the other name we call platelets. And you know what platelets do? Platelets are actually responsible for blood clotting. Okay, someone has an injury, now there's a cut, and then blood is what is already, there's bleeding. Okay, if you have your platelets active, working, thrombocyte, within a short time, what happened? The blood becomes clotted, that wound becomes clotted, and then there's no bleeding. The bleeding eventually stops. Okay, of course, that's why you discover that those that are diabetic actually do not have that, because their system has been broken, the immune system. So you see that when they have injuries, it is difficult for those injuries to get healed on time. Okay, because the blood clotting system has been, so the platelets has been paralyzed. So we have the thrombocytes. What they do is to enhance the blood clotting to avoid bleeding in humans. Because don't forget, we are talking about transportation of oxygen and carbon dioxide. Then option C say phagocyte. Phagocyte is a type of white blood cell also. Okay, that helps in the immunity of the body of the organism. The last one is option D, which is erythrocytes. The other name erythrocyte is called red blood cells. That's the RBCs, red blood cells. What do? Erythrocytes help in carrying, in transporting oxygen. Why? Because in that red blood cell we have a pigment that we call the hemoglobin. So the hemoglobin reacts with oxygen to form what we call oxyhemoglobin. Okay, so once that is formed, the oxygen is now being transported, okay, to the part of the body, through the heart, to where it will be useful. Of course, carbon dioxide is also transported and carried in the erythrocytes, which is the red blood cell, because of the presence of the hemoglobin, the red pigment that is present in the red blood cell. So, in other words, the examiner wants to know if you know the other name that the red blood cell is called, or what the function of the red blood cell is to transport oxygen and then carbon dioxide. Of course, the examiner expected that you should have known that the red blood cell actually transports oxygen and carbon dioxide, but they want to know if you know the other name, the scientific name that the red blood cell is called. Of course, they all sound the same. Site, and then we have site here. So erythrocyte is actually the red blood cell. So the correct answer is option D, erythrocytes. Question 19. The veins of the leaf are formed by the what? The veins of the leaf that we have in the leaf. Is it the vascular bundles? We have the cambium cells, the palisade tissue, the spongy mesophyll. Actually, the spongy mesophyll and the palisade tissue enhance photosynthesis, okay, to take place. They enhance the photosynthesis to take place. Of course, palisade tissue is also called the palisade mesophyll. Okay, so both of them are involved. The palisade tissue, what it does usually help to replace the dead cells, okay, that are present in the leaf. Don't forget, we are talking about the veins of the leaf, not the dead cell. So for the dead cell, the palisade mesophyll is responsible for that. It tries to replace it so that photosynthesis can actually occur and take place. Okay, the cambium cells also does that, because the cambium cells, what it does is they will help in replace the tissues of the cells, or the tissues or the cells, okay, in the leaf that are worn out. So that cambium does that, and it helps in maintaining the cells that we have in the leaf. But we are talking about the vein of the leaf. How are they formed? Where are they formed? It is formed by what? Usually it is what? The vascular bundles. Remember, we talked about the pteridophytes and the bryophytes being non-vascular bundles. So they will not have that taking place in them because they don't have it. But we have the pteridophyte, we have the gymnosperm, we have the angiosperm. Okay, those ones usually, because they have vascular bundles. Okay, that veins. That's why you see the angiosperm. You can see the leaves, okay, of the monocots, the angiosperm. They have their seeds, okay, enclosed in the fruit like that. You can see them that the veins of those leaves are formed usually by, you can see veins on the leaf. Okay, those veins on the leaf are formed in the vascular bundle system. Okay, so the correct answer here is option A, vascular bundles. Do you have any question bothering you at all on all we have done so far? Please feel free to ask. You can do that by simply clicking on the link in the description below that takes you to the Myschool website. There, you'll be able to ask your question and solutions will be provided within the shortest period of time. Now, let's go to question number 20. Like I said earlier on, that when you see a diagram first, identify what this diagram is, and then you'll be able to begin to ask the question. If you look at this, you can see something that is coiling like this, like a snail. Okay, that is the cochlea. Okay, we have some things here, then we have ear. Actually, this is the picture of the ear. Okay, and then how the sound is being generated and then heard eventually. So if you look at this diagram, we have this is, of course, this is the ear now, the outer part of the ear. Okay, that's the, we call it the pinna, the outer part, this part where you can touch now. Okay, that's the pinna, through which the sound will enter through there first. As I'm speaking now, you can hear me because the sound gets in through the outer layer, outer part of the ear, and then it goes through the ear canal. This is the canal. Go back figure 1. This is the canal. It is moving through the ear canal, and then as the sound gets here, the vibration will hit the eardrum. We have eardrum present at this side. So once it hits the eardrum will vibrate. Okay, once the eardrum vibrates, the eardrum now sends signal to some tiny bones in the ear. Now, don't forget that this is the inner part of the ear. We have the middle part now. That part, that's where the sound gets to, at the eardrum, where you now get the signal at those little three. We have about 3 tiny bones. Okay, we call them the malleus, we have the incus, we have the stapes. Okay, the 3 tiny bones, we get the signal from the eardrum, okay, and then send vibration straight to the cochlea. The cochlea, you can have it, the cochlea here, it is one that looks like the snail. You can see it like a snail kind of thing. Okay, once it gets that, then it gets the vibration and interprets. Then the cochlea now sends the sound, the vibrations, okay, message straight to the auditory nerve. The auditory nerve, of course, is the one that sends it to the what? To the brain, and then it interprets that, okay, this person is trying to do this person is trying to say this. Then you can interpret what sound that has gotten into the ear. So it moves from the outer part straight to the ear canal, from the ear canal straight to the middle part of the ear, okay, where we have the 3 tiny bones, the incus, the stapes, the malleus, and then that sends it to the to the inner part now, the outer, the middle, and the inner part of the ear, where we have the cochlea, and then it eventually sends the signal to the auditory nerve, where in the brain, where the brain can be able to work to. And so that means I say that the cochlea sends message to the brain, where it can be able to interpret what has been said. So now we understand what happens here in the ear. Now the question says, the parts which function together to bring about hearing are labeled what? Which functions together to bring about hearing are labeled what? Option 1, we have it here. Of course, that's the ear canal. The sound will get into the ear first and go through the ear canal. So option 1, we have it there. Okay, and then as it moves, we have option 2, we have 1, 2, 3. Okay, of course, seeing those 3, that tells you that is the 3 tiny bones. We call them the ear ossicles. Ossicles. Okay, ear ossicles. Those are the 3 tiny bones we have in the middle part of the ear. So it gets in there. Don't forget, there's the eardrum here first. But the eardrum is not actually. Then you have that in that part. So which is, it must get to the ear ossicles. That's the 3 tiny bones, which is ii. So that means ii is also part of it. And then it moves straight through this part, eventually, and then to the cochlea, the 3rd part. Now you can see it now, that moves into the what? We have the eardrum at this point, and then moving it that way to this part, which means, of course, there's no way it can, once it hits the vibration is sent eventually to this part, then it moves what? This way. So getting to I is also there, and then we have it moving to the what? To the cochlea. The cochlea is this round place now. That is what IV, okay? Okay, where it's eventually sent to the brain, the auditory nerve, where we have it. So V is not part of it. The VI is not part of it. So what we have is I, Roman figure 3, and Roman figure 4. So option A says Roman figure 4, 5, and 6. No. Roman figure B says, option B says Roman figure 1, 2, 4, and 6. No. Option C says Roman figure 1, Roman figure 2, Roman figure 3, Roman figure 4. Yes. And this is 1, 2, and 4. No. So the correct answer here is option C. Roman figure 1, Roman figure 2, Roman figure 3, and Roman figure 4. Now question number 21. It is still a continuation of the diagram we just dealt with in question 20. You can see that is still your ear, okay, human ear. We can have a Roman figure 1, that's the ear canal. We have Roman figure 2. You can see, if I want to see the 3 arrows, that shows you know what it is. That means it's pointing at the 3 tiny bones that we call the ear ossicle. So the question says the part labeled II, that's Roman figure 2, is the what? Is it the fenestra ovalis? No. Is it the middle ear canal? No. Is it the internal auditory meatus? No. We have the ear ossicle, option D. We said that earlier on, okay? The 3 tiny bones, 1, 2, 3, of course, that makes up that. That's the ear ossicles. The incus, the malleus, and then the what? The stapes. So the 3 of them actually make up the 3 bones that we have in the human. So option D is the correct answer. Now question number 22. When specimen X is mixed with few drops of iodine solution, the appearance of the blue-black color confirms that X is what? This is a confirmatory test for the presence of starch. You want to check if starch is actually present in the leaves, okay, that carries out photosynthesis. So all we need to do is we just get the leaf, you know the procedure, get the leaf, you wash it. When you wash it, you parboil it. When you parboil it, then a part that is there, you just cover some part, and then you put your few drops of iodine solution on that part, okay, and then you watch what happens. After some time, you discover that the color of that part where the iodine solution has turned to what? Blue-black color. So it is to check for the presence of starch in it. So option A says galactose, no, even though it is a carbohydrate. Starch, option B, yes. Sucrose, option C, even though it's a sugar, it's a carbohydrate, but it's not starch. Option D is glucose, which is also a monosaccharide, which is a type of sugar, which is also a carbohydrate, okay? But this is a confirmatory test for the presence of starch. So option B, because once you had it, the color will change to what? A blue-black color. So the correct answer is option B, starch. Question number 23. This is another diagram. You need to identify this. Of course, this looks like a bone, okay, of a skeleton. So, but you need to identify what part of the bone of the skeleton is this. If you look at this very well, this is looking a bit broad. We have these 2 parts, okay, and then you have this. Of course, this is where the spinal cord, okay, is definitely going to pass through, okay. We have this. So this actually is found at the neck region, okay. The spinal cord, immediately after the skull, don't forget that the skull, human skeleton, is connected directly down to the what? To the spinal cord. You can have it, going down like that. So we have the first part of the bone, human bone, from this part now, this neck region here, okay, is the cervical, okay. That's where we have this type of bone, cervical, and there are 7 in number. If you count, okay, if you have someone that can count for you, you can call the person. But if you see, have access to human skeleton, okay, you can just count from this thing. You count down like that. You have about 7 of them. You might not be able to see it, okay, but if you have the human skeleton, you can see it. So the cervical is actually found at the neck region, okay, of human skeleton. We have about 7 of that. So thereafter, after that, we have the thoracic, okay, the one immediately after the cervical, we have the thoracic. That is about 12 of that, 12 pieces. You can count 1, 2, 3. About 12 of that. Then thereafter we have the, okay, we talked about this, we have the lumbar, and then we have the chorda. Chorda has to do with that. The lumbar, we have about 5 of that also, okay. Immediately after the chest part, that's the the thoracic, you have that. Don't forget that these are bones actually on the spinal cord, the back, that makes it easy for you to bend, to stand upright. The positioning, okay, are being controlled. So the structure above actually represents, we have answered that already. This is the cervical, okay? That's the cervical vertebra. The thoracic is actually the one at the chest. The lumbar is found, and the caudal is found at the hip, the waist, like if the tail, okay, region where we have it. Immediately after that, we have about 5 of that. So the structure above, this structure actually is representing a what? A cervical vertebra. So the correct answer is option D. Question 24. This is the follow-up of what we just did in 23, the same diagram, okay? Still on the cervical vertebra. Now, if you look at this diagram, the question says blood vessel usually pass through the structure labeled what? Okay? This part, Roman figure 1, is the bifid, okay? That's where the spinous process actually takes place, okay? But it is not inside of this, okay? Then we have, there's this part here, which is this place here. Now you can see this part now. It's called, this part is called, okay, maybe I should just label this. This is your vertebral foramen. Okay, there are two foramen we have here. We have the vertebral foramen, and then we have this other one, like a cavity, like a hole, like that. Okay, and this other one is the other one, which is the transverse foramen. Okay, of course, this part is the body, okay, of the bone. We have the body, then we have the lamina. At this point, we say this is the bifid, where we have the spinous process taking place in this place. Now the question says that, don't forget, this is the what? This is the vertebral artery. Sorry, the vertebral foramen, and this is the what? The transverse foramen. This is the body. This is, now the question says blood vessel usually pass through the structure labeled what? Structure labeled what? Now the blood vessel will pass through what we call the vertebral arteries. The vertebral arteries, the veins, okay, actually pass through the same place where we have this. So the blood vessel usually will not just on its own flow, okay? It's a vessel, so it has to pass through the vein and then the vertebral artery, okay? Now the vertebral artery usually is passed through the transverse foramen, which is this part. You can see this hole here. This hole here, that's where we have the transverse foramen, okay? So that is where the artery, not this one, not this big one, not this vertebral one, the foramen here that we have here, that is where it passes to. So we can have the artery passing through this, okay? When we say the artery, we are talking about the vertebral artery, and it is through the vertebral artery that is where the blood vessels will usually flow through, okay? That's where the blood vessel will flow through. So the question now being asked that the blood vessel usually pass through the structure labeled what? Through this part. Now that is where the blood vessels will pass through. So which is Roman figure 5. Roman figure 5, that is V. So the blood vessel usually pass through the vertebral foramen, which is where usually the what? The blood vessel will pass through. So the correct answer is not Roman figure 3. It's not Roman figure 2, which is the vertebral foramen. It's not Roman figure IV, which is 4. But the answer is Roman figure 5. That is the transverse foramen, where the vertebral arteries, the vein, usually will run through, and the blood vessel actually pass through them. So the correct answer is option C, Roman figure 5. Question number 25. Salt and water are absorbed in the roots and transported to leaves by what? Okay, don't forget, salts and water usually are absorbed in the root ears, okay? Root ears, you have them, the root ears, and what? Absorb the water and the soil that are present in the soil, okay? And then it is passed through, the tree and through the entire plant. Now, once the salt and the water are absorbed in the root ears, okay, they begin to undergo what we call cell-to-cell, movements, okay? And then once they keep moving from one cell to another, from one cell until they reach the xylem. Don't forget the process through which that is done. The movement of the salt water through the air, root air, is usually through osmosis, because osmosis has to do with what? The movement of molecules or substances, okay, usually from a region of lower concentration to a region of what? Higher concentration through a semi-permeable membrane. Okay, that's what the definition of osmosis is. So if that takes place, as the salt and the water are moving through the root here from one cell to the other, from one cell to the other, it is going through osmosis. Okay, because we have that point, the concentration is lower, so it's taken to a region where it will be higher, where it will be really needed, where you have it much at that point. Okay, so once that takes place, it is through osmosis, and it will keep doing that. The salt and the water continue to move from cell to cell until they get to the what? The xylem. Okay, the xylem now transports that salt and water eventually into the other part of the disease, especially through the leaf, okay, where they are needed for growth and fat. So the question says salts and water are absorbed in the roots and transported to leaves by what? Diffusion. Of course, we have established that the process is through osmosis, okay, because the substances, the molecules of the water and the salts are in region of lower concentration, taking them to the region of higher concentration where they will be needed more. So it is not diffusion, because diffusion has to do with what? A movement of substances from region of higher concentration to a region of lower concentration, and not through a semi-permeable membrane like we have in osmosis. Okay, so through the xylem tissue, yes, of course, it's through the xylem tissue, but the process is not diffusion. So option A is not correct. Option B says osmosis, yes, through the phloem tissue. Yes, even though the phloem also, helps in transportation. Okay, we say we have the xylem and phloem. We call them supporting tissues. Yes, supporting tissues. Okay, from one part to another. But it is not the function of the phloem, okay, tissue to move salts and water. So the particle. Of course, there are other minerals that this phloem can also take, but not the salt and water. So this phloem makes this option B not to be correct. Okay, option C says diffusion through the phloem tissue. Of course, not diffusion. Process said it's osmosis, and it's not the phloem tissue. Okay, and then option D says osmosis through the xylem tissues. Okay, usually the cells begin to move the salts and water through osmosis from one cell to the other until it gets to the xylem tissue. The xylem tissue now moves them eventually to both the salt and water to the part where they are needed, growth and other metabolic activities and processes of the leaf. So the correct answer is option D, osmosis through the xylem tissue. I hope you enjoyed this content. If yes, please don't forget to click on the like button, hit the subscribe button, and finally tap on the notification bell to keep you informed once we upload our next video.